This Carla.
Eh, So, uh, can I start? Yes. Okay. Do you hear me well? Okay. Thank you. So, I'm Carla. First of all, thanks for giving me the opportunity to give this talk here today. I'm going to present the current work I'm doing in my PhD thesis that is titled Towards a Mechanism of RNA Polymerization and Non-Enzymatic Replication in the Early Earth, a theoretical computational approach. This is a work I've done in collaboration with Adrián Aguirre Tamaral, Jacobo Aguirre, and also Carlos Briones, that is an expert in the field of the origins of life in Spain. So, okay, this is my group where I'm doing the PhD thesis under the supervision of Jacobo Aguirre at the Astrobiology Center in Spain. And in our group, we try to understand how life could originate in our planet, but we know it's not an easy question, right? So we have to deal with a lot of uncertainties. And for example, one is that current biochemistry does not help us much. And why is that? Well, because it uses very complex mechanisms. Current living beings use these sophisticated molecules, such as enzymes, that have evolved over billions of years, right? However, at the beginning, life used much simpler mechanisms. So at the beginning, the environmental conditions played a key role in these origins of life, and probably aided by other available natural catalysts at that time, such as clays or ions or little peptides, etc. So we also know that it's very difficult to define what life is, but we can enumerate at least some of its fundamental characteristics. And from them, I'm going to focus here on replication. So with our model, we are trying to understand how the first information coding polymers emerged, and then we're able to self-replicate in this context of the primordial Earth, in which we didn't have complex molecules such as enzymes aiding in that process. Oh, yeah. And also, for this study, we are going to be modeling polymers and specifically RNA. And why is that? As the RNA world hypothesis highlights, RNA is a very interesting candidate for the origins of life because it has two very interesting characteristics. One of them is that it can perform catalytic functions such as our current proteins, and also it can encode genetic information in its sequence of letters, and it can also self-replicate thanks to the complementary of its nitrogen bases. And in our work, therefore, we are going to build a computational and theoretical model of polymerization and replication of these RNAs, and in a water-clay interface. And the clay is because it is known that certain clays favor the polymerization of RNA. In fact, this is a real image of the microstructure of a clay that is Mount Meridianite. And you can see that it is full of different channels of different sizes. In our model, we have divided our computational model into different compartments representing two different channels of this clay. In the first compartment, it's going to represent a very narrow channel in which just a single-stranded RNA can fit in. However, we have another compartment, that is compartment 2, in which RNA duplexes can form and can also fit in. I'm going to explain a little bit more in detail the computational model. Okay, in compartment 2, we are interested in studying random polymerization. We are going to have two different environments in the model, the water and the clay. By the way, this is a stochastic model. So we are going to start at the beginning with single ribonucleotides floating in the pool of water, let's say, and they are going to start falling into the clay. When they fall in the clay and they fall one next to each other, they can form a covalent bond, therefore forming longer and longer RNA polymers. These polymers that are attached to the clay have the chance to return to the pool with certain probability. This probability is the probability of desorption that will depend on the length of those polymers and also on parameter alpha. Alpha is representing the interactions between the clay and the ribonucleotide that are known to be non-covalent interactions that are weak, such as van der Waals forces. Okay. Also, so here we are going to be interested in studying what is the length of these polymers we can obtain by varying environmental conditions, that I'm going to explain you later. And also very important, we are going to take into account the length of those polymers just when they have returned to the pool, because we're assuming that those polymers can perform their functions when they are in the water phase and not when they are attached to the clay. And there is an example of a single simulation and how our model works. In the y-axis, it is displayed the position in the clay that we have modeled as a unidimensional array. And the clay can be occupied in blue or not over time. So you can see the kind of polymers that start to form. When they disappear is because they have returned to the pool, and so on. And now I'm going to pass to explain compartment 2. Simulations are very similar, but here we are interested in studying replication. Because here we want to start replication, we start our simulations with an already formed polymer that in this case, we're going to use 20 ribonucleotides long polymer, because with 20 nucleotides, these RNAs can already perform some very simple functions. Therefore, in this case, we want to replicate this initial polymer that at the beginning, it's going to be attached to the clay. Here, random polymerization can also take place, and exactly in the same way as in compartment 1, with parameter alpha and the desorption probability of all the molecules attached to the clay. However, here, ribonucleotides can also fall on the top of the original sequence of the template. And if they fall on the top of the template and the nitrogen bases are complementary, they are going to start forming a complementary copy of the original information. This complementary copy can denature from its template and reattach elsewhere on the clay, and perform again another round of complementary copying, therefore forming a replicate. The replicate, it's going to be a sequence that is identical to the original one, or at least to a part of the original sequence. And when again these replicates are returned to the pool, is when we are going to measure their lengths. The length of these replicates is going to give us like a proxy of what is the efficiency of the replication of this original information. Okay. So here, sorry. We also have another important parameter that is beta. Beta stands for the interaction for the hydrogen bonds between the ribonucleotides. And therefore, each RNA duplex is going to have a probability of denaturation that will depend on the value of this beta and on the length of the duplex. And here I've also put a single simulation. So you can see it's very similar to compartment 1. However, we have now different types of polymers depending on the color. We are representing the original polymer that is attached to the clay in light blue, for example. Different colors stand for complementary or for complementary copies of the original. And also when a position is filled with a double-strand RNA, it is colored in purple there. But important, how do we introduce the changes in the environment, right? Because this is what we want to study. We are going to introduce them as changes in the values of these alpha and beta parameters. We can think about them as the strength of these two kind of different interactions. Therefore, in a particular environment, these interactions are going to be more or less stable. And this is how we are going to model the environment. Therefore, if we are in a very harsh environment that does not favor polymerization, we are going to have small values of alpha and beta. And otherwise, in a build environment, they are going to have larger values. And now I'm going to pass to the most important results we have obtained. Okay, there there is like scientists are starting to realize that it seems that fluctuating environments in the early Earth were important for the emergence of life somehow. So we try to compare what is the difference in polymerization and replication, comparing constant versus different fluctuating environments. And we found that both oscillating environmental conditions are very important for the formation of longer RNA polymers in compartment 1, and also they are essential for obtaining replication in compartment 2. We model different kind of fluctuating environments, for example, random fluctuations, or sinusoidal-like oscillations with different periods, different amplitudes, also square-like waves, let's say. And yeah, also to highlight here that for replication to take place, the most limiting factor is beta. So changing the value of alpha, so those clay ribonucleotide interactions seem not to be very important for replication, but beta seems to be a very important factor. So you can see here that when the value of beta is constant, remember that the initial polymer we want to copy is 20 nucleotides long. We are not obtaining quite good results, but for certain values of this beta, we obtain more efficient replication, and even we obtain entire copies of the initial sequence. Okay, so now we arrive here. We know that we have tested with our model at least that fluctuating environments or oscillating ones are very important or are key for replication. But what kind of oscillating environments? Can we say something about or point towards certain environments in the early Earth, more likely than others? Okay, so we here perform an analysis of the different values or parameters of the oscillation. We have taken sinusoidal oscillations for simplicity. And here what is displayed is a scan of some parameters of the oscillation. So in X and Y axis is the central value of the oscillation, and each heat map is represented a different relative amplitude of the oscillation. And the most the thing we want to highlight here is that it seems that for replication to be efficient, we need very strong environments. So we need that the amplitude of the oscillations we are modeling is very high. That could be interesting because if we go to real environments, let's say in the early Earth, it could mean that certain environmental changes, periodic environmental changes, such as, for example, day and night temperature cycles, may be too soft for this kind of replication to take place. However, other more drastic environmental changes, such as wet and dry cycles, could be more promising. Okay. We have also modeled how the change in the period affects replication. And we have found that not all periods are suitable for replication. It's also this maximum depends on the parameters, but it's there. And it also sounds interesting, although in our model, we are trying to currently link one time step of our model with real time because it doesn't represent it right now, but we are working on that. Is a period. Yeah, that's what I'm saying. In our model, the time step is not related with real time. It's one time step of our model. Yeah. It's the replication. So no, one time step is just one time step is represented by we choose one molecule from the water that falls into the clay. This is one time step. And all the molecules that are attached to the clay or the duplexes can also be the sort of the nature in that time step. This is what it means. So we are currently trying to link this with real time. Okay, but we have also looked at what is the real time that it takes for an RNA to replicate. And we have found in experimental data that the rate in this kind of conditions is about you incorporate one nucleotide every 10 to 4 or 10 to 5 seconds. Therefore, non-enzymatically, of course. Therefore, if you want to replicate a 20 ribonucleotide, you will take around 2 to 20 days. This is the range we have calculated. And if we think in this kind of period in the early Earth, we can think about spring tides, for example. So because at that time we have calculated that they last around 7 days. And it is interesting because if these calculations are okay, that maybe not, it could be highlighted a role of the moon in this kind of replication dynamics. Okay, finally, we also studied the role of the genetic alphabet in replication. We could do that with the computational and with the analytical model. Because it is still a very intriguing question, why our genetic alphabet has 4 letters, right? Therefore, we here compare different alphabets of different sizes around different periods of these oscillating dynamics. And when we compare them, we find that the smaller the alphabet, it seems that the better is the maximum replicates we can obtain. So this in principle is pointed at smaller alphabets that will be more better in this term, although there is for each period, there is an alphabet that is better than the others. That is also interesting. However, we know that it is not all the length of the replicates, but there are other important things. So we compute a very simple measurement of the molecular diversity of those molecules. That is just the number of different sequences we can make of with a certain alphabet of and a molecule of a particular length, and that is the value of the molecular diversity. And when we calculate that with those replicates lengths, we find that it seems that 4-letter alphabets with that seems to be an optimum in this kind of molecular diversity measurement. What is interesting, because our alphabets are 4 letters, and if we compare also a 4 canonical alphabet with a 4 vowel one, and that is just our current alphabet is 4 vowel because you have the canonical interactions that are guanine with cytosine and adenine with uracil, but another less stable one that is, for example, guanine with uracil. If we introduce that, it seems that this kind of alphabet really outcompetes the rest. However, we need to be careful because this also introduces mutation that at the beginning is not going to be fine. And just I'm going to summarize this because I'm running out of time. So we also built an analytical model, but I didn't have time here to explain. But just to tell you that it recovers the phenomenology of our simulations, that it is a simpler version of the computational model, and most importantly, that it allows us to demonstrate that oscillating environments are always better than constant ones for replication. Also, same trends with period, amplitude, and same conclusions with the alphabets can be done. And yeah, finally, these are the conclusions. Let me just summarize that we have built this computational model of polymerization and proto-replication just driven by environmental forces in this water-clay interface, that it may allow us, this study, to narrow the amount of habitat suitable for the emergence of life in the early Earth. We think it is also a powerful tool to test multiple conditions compatible with with prebiotic world. That 4-letter alphabet seems to outcompete the rest when looking at molecular diversity. And finally, that we have also developed this analytical framework I've just told you about. So that's all. Thank you very much for your attention. And any questions?